Good morning, everyone. I would first like to thank the organizers for allowing me to speak today. And before I begin, I would like to disclose that I do not have no conflict of interests. And I do also want to disclose that the work I'm presenting today is not of my own. So to begin, I would first like to introduce the intestinal or the human microbiota and its roles in host physiology. Next, I will discuss the environmental influences on the microbiota and the current literature on non-caloric artificial sweeteners and how it may be involved in altering the intestinal microbiota and host physiology. I would then want to change gears and discuss how to design a microbiome study and how nutritionists should really try to jump in on this field. And then finally, I would like to summarize current literature on the intestinal microbiota and in the context of non-caloric artificial sweeteners and give some of my thoughts on future directions. The human body is colonized with over 100 trillion microbial cells. These microbial cells represent 3% of the body's mass, and the conglomerate of these microbial cells outweigh host cells by a factor of 10, and their genes outweigh host genes by a factor of 100. These microbial communities are niche-specific, and they have substantial intra- and intergroup variation at different anatomical sites. The intestinal microbiota is acquired very early in life, at birth specifically, and these communities are low in diversity and richness, and through ecological succession, these microbial communities stabilize, and by age 3 of life, these communities are stable and remain relatively stable throughout the life of the host. The intestine harbors over 10 to the 12th microbial cells, and these microbial communities are influencing intestinal and host health, including, and they provide protective, structural, and metabolic functions to the host. On the immunology side, the microbiome is known to support host immunity. It's involved in host immune homeostasis, included by the production of polysaccharide A and the induction of regulatory T cells, or metabolizing polysaccharides, producing short-chain fatty acids, which then in turn can influence the induction of regulatory T cells. And finally, through lipopolysaccharides, we can see an activation of pro-inflammatory cells and the maturation and education of immune cells. The microbiome is also involved in protective immunity. It provides competition against pathogens through competition for nutrients, and also is involved in the production of antimicrobial peptides. The microbiota is also known to alter host metabolism through both diet-independent and diet-dependent factors. On the diet-independent side, the lipopolysaccharide is known to induce insulin resistance through the induction of toll-like receptor 4. And peptidoglycan is also known to induce insulin resistance through the induction of NOD receptors. Diets rich in choline are known to be metabolized by the host microbiota, producing trimethylamine oxide, which is known to be involved in cardiovascular risk. And as I mentioned before, polysaccharides are known to produce short-chain fatty acids, are metabolized by the microbiota and produces short-chain fatty acids, which can regulate energy source and induce the expression of G protein-coupled receptors that can influence the regulation of PYY and GLP-1. In the center of this diagram are microbial products and activities that are regulated by the intestinal microbial community. These microbial communities are in a delicate balance between health and disease. And certain perturbations may actually alter this delicate balance and shift the host from a healthy state to a diseased state. Certain factors that are involved in shaping host microbial composition include antibiotics, lifestyle, diet, and hygiene, as well as having a hyperimmune response or alterations in genetics. These altered microbial community state, or this dysbiosis, can be involved in promoting chronic inflammatory responses, autoimmune diseases, or metabolic dysfunction. The question arises, does the non-caloric artificial sweeteners have an effect on the intestinal microbiota? And if so, does it have an effect on host physiology? The FDA has approved 6 artificial sweeteners, which are involved in regulating host weight and blood glucose levels. Today, I'm going to present to you 3 papers that have highlighted the effects of aspartame, saccharin, and sucralose on the intestinal microbiota and its potential effects on host physiology. Palmas and colleagues gave aspartame to Sprague-Dawley rats in the context of a normal chow diet or a high-fat diet. And in the rats that received aspartame under a normal chow diet, you saw a decrease in total bacteria, an increase in Clostridia leptum, and an increase in Enterobacteriaceae. Therefore, they were able to define alterations in the intestinal microbiota, and this was using 16S quantitative polymerase chain reaction, using primers that were specific to specific taxa, including Bacteroides, Bifidobacterium, Lactobacillus. In addition to profiling the intestinal microbial community, this group also associated the dysbiosis with multiple effects on metabolic health. There were decreases in total weight. However, there was increases in blood glucose levels. Next, the Abudonia and colleagues asked the question, what was the effect of sucralose or Splenda on the intestinal microbiota, and what are the downstream effects on physiology? And to answer this question, this group also used Sprague-Dawley rats, and they gave a Splenda treatment at 100 milligrams per kilogram, 300 milligrams per kilogram, 500 milligrams per kilogram, or 1,000 milligrams per kilogram. And they gave it to them for 12 weeks, and then another group of mice had their Splenda water removed and were allowed to recover for an additional 12 weeks. So during the Splenda treatment period, there were decreases in total anaerobic bacteria, Bacteroides, Lactobacilli, and Bifidobacterium. All these bacterial taxas are involved in regulating host metabolism and immunity. During the recovery phase, there were also alterations. These alterations, some of these communities did not return. There were still alterations in total anaerobic bacteria, Bacteroides, and the levels of Bifidobacterium remain low. These microbial changes were also associated with elevated expression of P-glycoprotein and the members of the cytochrome P450 family, which are involved in xenobiotic metabolism. Most recent paper from Ruiz and colleagues asked the question, what were the effects of saccharin on the murine microbiota? So to begin, they first gave either aspartame, sucralose, or saccharin to mice for 11 weeks. However, the strongest effect was seen in saccharin, and therefore they proceeded to use this group to study alterations on the intestinal microbiota. And what I'm showing you here are called principal coordinate analysis. Each dot or triangle is a microbial community. So each dot represents one mouse's microbial community. And the circle, the black circle, triangle, square, and hexagon represent either water controls, sucrose controls, glucose, or saccharin controls at baseline or at week 11 post-saccharin treatment. And the blue triangles represent the mice that received saccharin treatment at 11 weeks of life. And what you can see is the distinction between the blue triangles and the black, suggesting that there were alterations in microbial communities. Next, they asked the question if these communities are transferable. And what they did is that they took fecal contents from either saccharin-exposed or control mice and gave them to germ-free mice, asking the question, are these communities transferable? And what you can see is that there is this distinct separation between the antibiotic, the saccharin-treated groups or the control groups, suggesting that these communities are transferable. Unfortunately, no statistical analysis was performed. Next, they looked at the composition. We saw differences in diversity. So next, what is being changed? So the red boxes correspond to taxas or microbial populations that are elevated with saccharin treatment. And the green boxes represent microbial taxas that have decreased with saccharin treatment. And with saccharin treatment, you saw an abundance of in the order Clostridialis, Ruminococcaceae, Lactobacillus, or Bacteroides. And with saccharin treatment, you also saw a decrease in Bacteroides genus. This could just be other microbial taxas, other species that in that genus that have been changed, and as well as a decrease in Lactobacillus reuteri and AF12. Next, they used shotgun genomics to look at the microbial genes that have been altered. And what they determined, there were several genes involved in the glycan degradation pathway that were increased. These genes are involved in the production of short-chain fatty acids, which would increase the amount of nutrients available to the host. And then finally, they wanted to know if giving human subjects saccharin would alter their glycemic responses, and also see if they would see any changes on microbial communities. And what they did is they gave 7 subjects, 5 males and 2 females, saccharin at 5 milligrams per kilogram per day. And then they measured the glycemic responses. So there were 4 out of the 7 that actually had increases in glycemic responses, and the other 3 did not have any responses. On the right here are the microbial community analysis between the responders and the non-responders. And this suggests that there is some sort of difference between the communities of the responders and the non-responders. And here you see the taxa abundance plot at day 1, where the ones that did respond to saccharin already had an altered community, suggesting that this altered community may actually make them predisposed to saccharin-induced alterations in physiology. What really was really interesting and exciting that they did was actually transfer fecal samples from these subjects into germ-free mice, really trying to link causation. And what they did was they took either samples from a responder or non-responder and gave them to germ-free mice. The mice that received fecal samples from the responder had an increase in glycemic responses, as shown here, while the mice that received fecal samples from the non-responders you didn't observe any changes. And when you look at their communities, they look pretty similar to the humans, to the human subjects. And their microbial communities do not change with the non-responder group. And in the responders, you see alterations in certain taxes. So this work suggests that saccharin-mediated effects on the microbiome of host physiology are transferable to mice. However, there are always limitations in these studies, and I just wanted to highlight some and something to take into consideration if one is interested in performing a microbiome analysis in a nutrition study. One limitation is dosing. Some of the doses that were used in these studies were not comparable to what humans consume. Next were the models. Are rodent models an appropriate proxy to human biology and human physiology? And it's something that all scientists that use a rodent model question and think about. And therefore, it's important to do both incorporate human subjects in microbiome work. Next is study design. Make sure you have a sufficient sample size and sample population, because that can affect statistical analyses and really decrease the power of the work. Next is the approach on analyzing microbial communities. So there were three different methods that were used to analyze microbial communities that I've presented. One was qPCR, using primer specific to certain communities, to certain bacteria. They were using cell culture, and the final one was using deep sequencing analysis. Both culturing techniques and qPCR provide a limited microbial survey of the total community structure. Therefore, I suggest if one is interested in looking at the totality of the microbial community and how it's affected by artificial sweeteners, one should use deep sequencing and analysis. And if anyone's interested in looking at the function, then culturing should be an approach that one should do. And finally, there were not that many statistical analyses performed on understanding dysbiosis. So we saw before in the PCOAs, we saw some differences between the responders and the non-responders. It would be nice to see that they're significantly different. So next, now I'm going to just discuss how to design a microbiome study and what to take into consideration if one were to design try to understand the effects of low-caloric sweeteners on the microbiome. One can use rodent models, and the advantages of using a rodent model is that the availability, the fact that you have both inbred and outbred strains, transgenic mice to understand the effects of host genetics, and the fact that you have this germ-free system where you can give whatever microbe that you would like to give and see its effects in the context of low-caloric sweeteners. However, you do have certain cons, including maternal effects, cage effects, and how the environment may influence the microbial community. Human studies, they're biologically and physiologically relevant. However, the sample accessibility and obtaining the proper controls to measure the gut microbiome may be very difficult. And then one should also take into consideration how samples are stored, extracted, and how the preparation is performed. And it's really important to mark everything that is performed in the study, document all covariates that may be factors in altering microbial communities. And then finally, when you perform a microbial analysis, make sure you consider diversity, both intra and interdiversity, alpha and beta, community structure, as well as using modeling and network analyses to truly understand how microbial composition and dysbiosis may be a factor in the physiological responses we see with non-caloric artificial sweeteners. So in summary, I would like to say that the intestinal microbiota is an integral part of human physiology. Environmental factors are known to influence community structure and function, and what these 3 papers I presented have shown is that aspartame, sucralose, and saccharin alter intestinal microbial community composition and structure. Additionally, microbial dysbiosis may be associated with alterations in host metabolic profiles and xenobiotic metabolism. However, further studies need to be performed by experts in nutrition as well as experts in the microbiome field. So future directions, as I mentioned before, further studies, further clinical and rodent studies are required to define the long-term effects of non-caloric artificial sweeteners on the intestinal microbiota, and one should try to define the mechanism by which these interactions occur. With that, I would like to thank you for your attention.